between processes extending here, there, whatever. Okay, so if you take it, a section like that and show everything, you would probably see profiles of an axon here, maybe a dendrite there. So these guys are enveloping not just one neuron, but a lot of them. Okay, that whole region. And so they must do other things. Right? They somehow must be controlling things around neurons that might be important. And we'll talk about that in a second. So not just holding. So in the central nervous system, you have those things. You have astrocytes, and you have oligodendrocytes, which is maybe business for cell, and you have microglia, and you have ependymal cells. I'm not sure exactly what they do, and they're not very studied. But these guys are distinguished histologically at the tissue level because they contain a lot of an intermediate filament, similar to the one I mentioned, intermediate filaments, the neurofilaments that are mostly made of proteins that are derivatives or very similar to keratin, what our hair and nails are. But these guys have one that's called gliofibrillary acidic protein. I don't expect you to remember this, but they can be identified very nicely. Because there is one thing that they express a lot of and very few others express a lot of. Oligodendrocytes do something else. They produce myelin. Talk about myelin a lot more. Microglia are tiny and they move a lot. They don't move a lot, but they can move. And they act a little bit as macrophages of the central nervous system. So they engulf things and they clean up. So they're kind of the vacuum machine of the central nervous system. Just right for that. And the general cells that you don't know what they do. I didn't even know about this. Oh, I see. Yes, I remember now. They make up the walls of the ventricles within the brain. You know, the brain has 4 cavities within that produce the cerebrospinal fluid that fills the interior, the center part of the brain, and the ependymal duct, which is the center of the spinal cord. So the spinal cord has a hollow duct in the center that's filled with fluid. These guys line all those compartments and probably are involved in producing some of that fluid, but I really don't know, and I'm not going to know. Okay, so walls of ventricles and okay. Now, if you look at this, what do you think that is? What it says there. Now, see it well? What do you feel about that? Now, what do you see in the structure of this thing? There's something very special about astrocytes. Yes. It connects to blood vessels. Yes. Well, yes. It has those little terminal blobby things that are called emphy. These guys. So the picture that I showed before, the diagram didn't have that, so that it wasn't a very good one, but it shows the starry nature of these things. But these guys are very important because they have these, and these emphy connect to the two blood vessels. And on the other side, they are all over and near the surface of the cell of the brain. And they penetrate further into the nervous tissue, to the brain. So the neuronal tissue, really, not just the brain. So what do you think they do? What does this suggest?
They probably carry nutrition.
Very good. So, okay, that's one thing. What else? What else do you think they might be doing?
anchoring other neurons and blood vessels around them, so they're in a fixed position
Yeah.
for like structure wise.
Yes. Yes, they do participate in that. You know, I really don't think that this is the main function of glia, although that's what they were initially supposed to do. And so in terms of anchoring, yeah, sure, they participate. But there's a lot of other matrix tissue and so on around neurons and so on that hold them in place. So although they certainly do provide mechanical stability to the thing, I'm not sure that really is that crucial, but sure. Yes?
Dilation for the axons?
No. Astrocytes don't.
Did you say like constriction and dilation of the blood vessels?
They regulate constriction and dilation of blood vessels. That's a good point. Maybe. You caught me there. Maybe. I don't think so, but maybe. So that's something to look into. Create a barrier. Yes, they do create a barrier in the sense that they are the intermediaries between blood vessels in the central nervous system. So yes, they do create a barrier. You've probably heard of the blood-brain barrier, which is a very tight seal, if you like, between the bloodstream, circulatory system, and the nervous system, so that very few things can cross. And that's why it's very rare that a virus will invade the nervous system. And when it does, it's extremely hard to get rid of it because chemicals won't go in there. Another very well-known example is dopamine. Dopamine is needed in the central nervous system, and it's depleted in certain important structures, for example, in Parkinson's disease patients. But you cannot inject dopamine in the bloodstream because it doesn't cross this barrier. However, the blood-brain, and that's why you inject L-dopa instead, which is that's crossed, and that gets transformed into dopamine inside the nervous system. But astrocytes don't really make up the blood-brain barrier. It is a barrier to the flux of certain things, but the tight seal of the blood-brain barrier is really produced by endothelial cells of the blood vessels, which are very, very tight, and whose tightness is regulated. Okay? So they can let things in. That's how they make hormonal regulation and other things. Maybe also by astrocytes as what's your name again? What was your name?
Miguel.
Huh?
Miguel.
Yeah. So Miguel says. So that I need to check. What else do you think astrocytes do? So they bring food or nutrients, as Emma said.
Oxygen?
No. Oxygen crosses and then makes food.
I guess if they bring food, that means they also get rid of certain waste, like
Exactly.
excess
And
potential.
it's an extremely important function. So it acts as the intermediary or the regulator, the valve, if you like, for transport of nutrients in and waste materials out of the nervous system. Okay? And that's a very important one, and I'll tell you a little detail in a second about it.
So, do the microglia, are they the ones that get rid of the waste, or is it the astrocytes?
No, microglia, not the same ways. Microglia engulf bigger things like dead neurons, bacteria, viruses. If they get in there, they really act as the macrophages. You know, macrophages are these phagocytizing.
So it'll be like the immune system of those nerves?
It's part of the, yes, and they have a different origin too. All these cells, these astrocytes and oligodendrocytes, have the same origin, developmentally speaking, as we become, go from an embryo into adulthood. These cells derive from the same tissues as neurons do. While microglia derives from mesoderm, which is what blood cells and other muscle and so on, other tissues also derive from. So they have a different origin, but they look a little bit like these glia. They're localized there, so that's why we categorize as glia, although they don't have the same developmental origin. Nobody else? No? So here's an important thing. I'm going to take a little jump and then later come back to this. When a neuron is active doing things, as we will see very soon, it does, it produces changes in voltage across the membrane that then are transmitted along the dendrites and axons. Okay? So that voltage and how that is produced, we're going to focus on next, but the way electricity is carried in the nervous system is how? Do you know? I mean, in the wires that feed all these machines and lights and so on, what you have is metal wire and electrons moving along the surface of the metal. Right? So that's what carries electricity. The charge that moves very freely on copper wires or metal wires in general, is what carries electricity there. What do you think carries electricity in cells in general, and neurons in particular?
Ions.
Ions. What are ions? Or charged atoms, right? So they have a charge also. They can be positive or negative, right? Electrons which are only negative. So, but they carry a charge, and that's an important thing. So when an ion moves, let's say a sodium or a potassium ion moves in space, it carries current. And it's an electrical current, just like the electrical current on these wires. It's just that it obeys laws that are slightly different, but not that different. In general, it's exactly the same thing. They move a little slower, perhaps, because the material they're moving in is different, and so on. Water. But that's what carries current. Now, in order for this to happen, they need to move. And they need to move from the inside to the outside of cells or backwards, across the membrane, the cell membrane of the neuron, the cytoplasmic membrane of the neuron. Now, when a signal is sent, typically a lot of potassium moves from inside to outside. Now, imagine this. You have a cell. I'm going to draw it round like this, with a lot of potassium inside, and not a lot of potassium outside. That's a fact. In our bodies, neurons, as well as other cells, have more potassium inside than outside by a factor of about 10 or more, right? So when there is a signal, one thing that happens is that sodium moves in, we'll talk about that, but potassium moves out. Now, the way I'm talking about these cells, and I'm including dendrites, is that these cells, and this is a neuron, these cells are enveloped by other cells. And let me draw more than one here. Okay? And that leaves a tiny little space here between the two cells. This extracellular space. So this stuff does not move there, it moves here. What is the consequence of that, do you think? What could be the consequence of something like potassium moving into this narrow extracellular space? Put it this way. What defines concentration of a substance? Okay, so and you're on the right track, but let's go back to concentration. Concentration is the quantity of something that we call a solid in a volume, right? So it's something divided by volume. Mass, expressed in whatever terms, divided by volume, right? So if the volume is small, as in this case, you can change the concentration there very readily because the mass will move into a tiny volume. It's not diluted in a huge volume. It's diluted in a tiny volume thanks to the fact that the nervous system and all our organs are packed so tightly with cells. That is a humongous problem because if potassium moves out from where it's very concentrated to where it's not, in between here, it's due to diffusion, as you would probably already guess, right? So down its concentration gradient. But if it starts accumulating here in a very tiny volume, that gradient is going to dissipate rather quickly. And it does. In fact, the concentration of potassium when there is a lot of neuronal activity concentration of potassium in this extracellular space or intercellular space grows quite rapidly. So you need to get rid of it. And that's one of these things, one of the things that astrocytes do. Very, very important one. It's called buffering, extracellular buffering. So that's one of the main functions, extracellular buffering, because it really acts as a concentration buffer, like chemical buffers, like pH buffers don't allow pH to swing wildly. These guys, by a very different mechanism, don't allow potassium concentration in other ions to concentration to swing wildly in this extracellular space. Okay. Crucial. Without that, if you kill astrocytes, the nervous system falls apart entirely. And there are cancers, for example, that can target astrocytes, and of course produce quite severe problems. So how do they do this, by the way? By the same mechanism as the mechanism by which potassium moves into this extracellular space. The driving force, what drives the potassium to move, may be into this cell, in the concentration here as well. So if you have a cell, and I'm going to draw it very simply with one end put here, the soma, and another one there, and let's say this is your capillary here. If this cell if one cell is sitting up here and producing it and being very active,